The Green Campus Enterprise Aquaponics and Bioretention teams design Expo submission for spring of 2024. Green Campus's mission is to annually ensure the carbon footprint of Michigan Tech campus and design and implement projects to improve the sustainability of Michigan Tech. The Aquaponics team members consist of Maddie Krasuk, Joey Gannon, and Ray Bensley. The aquaponics team objectives. Aquaponics is the agricultural relationship between fish, plants, and bacteria. Growing fish and plants symbiotically is an example of sustainable agriculture. Because of the reduction of soil erosion, land use, pesticides, fertilizer, and water use compared to traditional farming. Our goal in this area is to increase knowledge on this topic and spread it to other universities and outlets for education. Our on-campus specific goals for our systems are to use a system as an educational tool for classes such as microbiology, botany, water treatment, and fish biology. We also hope to use it as an experimental tool to measure different setups, different feeding regimens, and plant use, and potentially even fish use. We also hope to provide fresh vegetation and herbs and fish for local students. The general aquaponics layout can be broken up into 5 main steps. The first being to feed fish in your fish tank. This nutrient water is then pumped into an intermediary tank called a sump, where it is mixed with runoff from your grow bed into a half and half slurry. This slurry is then pumped back into the fish tank and to the grow beds. From the grow beds, the nutrients in the sump tank water is weeded out using nitrifying bacteria, and is then drained back into the sump tank where it's mixed with more water from the fish tanks. We have 2 main system design locations, the first being Dow 218. This is a larger floor plan, allowing us to have a significantly larger system. It's good for experimental use with a footprint of about 100 to 120 square feet. We are hoping to set up a mirrored system of 200 gallons apiece with 3 grow beds. This would allow for a split of 100 fish throughout the system total. The second location is the bubble greenhouse. This is a significantly smaller system, only about 180 gallons, leaving room for about 50 fish. It would consist of 2 grow beds and 1 tank. This would be more of a display unit and a teaching tool, less likely to be used for experimental use. Aquaponics prep work. A lot of prep work preceded this semester for the aquaponics team. Most notably was an estimated equipment cost for a brand new system. Both a maximum and a minimum cost were analyzed, the minimum being just over $1,000 and the maximum around $4,500. An electrical load and cost analysis was also performed on 3 various system sizes for the lighting in the system and any pumps that would be used. It is most likely that a medium system will be used for our operation, which uses about 2,041 kilowatt hours a year and costs just under $130. Fish procurement was also very important. We decided to choose Americulture as our primary source of tilapia. They met all of the in-state requirements for disease and health control for their fish and had the proper paperwork to back it up. Additionally, we were also able to model a nutrient cycle for an aquaponic system to get us a good head start on how to set things up properly. Ethical concerns. In order to ensure humane treatment of the fish, the team wrote an IACUC protocol and took CITI training. This training consisted of animal biosafety, animal care and use, working with fish in research settings, and working within IACUC. The team gained insight on ethical euthanasia, husbandry, and our own personal safety and PPE techniques required for handling fish properly. When it came to the IACUC protocol, we also spent a lot of time researching different ways to refine, reduce, and replace possible areas of harm for our fish in the system. Our next steps going forward are very simple. We plan on buying equipment, assembling our system, and finally transitioning into an experimentation period on one or both of the systems we have available. We also hope to continue work with prospective projects. Keweenaw Cape is interested in future partnerships. Additionally, the Waters Meat Trout Hatchery is interested in a working relationship going forward, looking for insights on different ways that they can implement an aquaponics system on their property. And we hope to advise both going forward.
The bioretention team is working towards utilizing bioretention systems like rain gardens or bioswales to move and treat stormwater in areas where stormwater accumulation is an issue on campus.
For this project, we have been fortunate enough to work with Quincy Arney, Michigan Tech's Master Gardener. She has assisted us in getting permission to test the soil on campus and has guided us in site selection and with some of the more creative aspects of our design work.
The team consisted of Aaron, Allison, myself, and Gibson, who are all senior level environmental engineers.
Last year, the team conducted desktop research and created a map of areas on campus with stormwater issues. These 3 sites were chosen based on the amount of foot traffic and the amount of water that accumulates during snowmelt and rainfall events. The 3 on-campus locations are outside of Fisher Hall, outside of Walker Hall near Highway 41, and by DHH.
At each site, we performed a percolation test in addition to collecting soil samples to conduct a grain size analysis. In bioretention, a design's ability to process stormwater is affected by its grain size distribution. This is related to hydraulic conductivity and soil compaction, as too high or low values will inhibit water movement and stability of the system. The grain size analysis was completed using a tower of sieves, which tells us how much of each size particle is in each sample, and what amendments might need to be made to enable correct function of the system.
We analyzed possible sites using ArcGIS and rainfall data provided by the Geospatial Research Facility by creating a pour point at the spot of highest accumulated flow and using the watershed tool to determine the length and area of the drainage catchment. We were also able to determine soil as Class B and land use as urban and built up across campus. Walker had the largest drainage area, collecting water from both Walker and DHH. Because of its size, especially in comparison to the other 2 sites, we decided that we would prioritize this location for implementation because it has such a high foot traffic and because it's only going to get busier with the addition of the new dorm. Fisher and DHH had very similar drainage areas and lengths, differing only by 22 feet squared and 16 feet.
To calculate the volume of the rain garden needed, an Excel sheet using the Soil Conservation Service Method, or SCS method, created by the Michigan Department of Environment, Great Lakes, and Energy was used. This method is used for flood discharges for small, ungauged watersheds, which is what the watersheds on campus are. The SCS method makes a dimensionless curvilinear unit hydrograph to show the amount of precipitation that goes into the basin outlet, or in our case, our pour point. We determined the volume under the hydrograph is the total volume needed to collect the runoff of a 100-year, 24-hour storm event. The hydrograph shown on screen is for our Fisher Hall location. The calculated volumes are used in the final designs at each of our locations.
For the Fisher site, the final design accounts for 357 cubic feet of water collected over the drainage area. The garden area will be 526 square feet and 5 inches deep, with a 3.2-inch berm on the south side to collect overflow. For the DHH site, 358 cubic feet of water will be controlled with a 512 square foot garden, 5 inches deep, and with a 3.4-inch outer berm. For the Walker site, due to the large volume of water draining to this point, any garden installed here would be undersized for a 100-year, 24-hour storm. Assuming a ponding depth of 5 inches and a berm height of 4 inches, even with the maximum area available, a garden would only control about 60% of the runoff. With the practical constraint of a 500 square foot garden, only about 2% of the runoff would be controlled.